Okay, another tool I think is very unique is this Busca. So this tool is actually an example of many tools. So um, this Busca integrates different tools to predict the protein localization, including DeepSeek, TP-Pred3, Pred-GPI, and the Beta-Aware, and example 3. Also, some globular and membrane proteins prediction tools, including Barcelo, MAM loci, and S. Chlora. So you can also see the tools integrated here. All the tools are developed by this group. So it is a group in Italy, and similar to another group we know in Europe, they kind of developed their own ecosystem here. So you see this Pusca is for general protein localization prediction, and also they have this DeepSeek that is specific for uh, protein signal peptide. And this TP-Pred is for target peptide predictions. And this Pred-GPI is for GPI anchors on protein cell surface. And also this one is also a general protein localization prediction tool. Asclora is a sub organelle prediction tool for proteins in the chloroplast. Beta-aware and mamloci are tools for protein localization predictions specific on the protein membranes. So this one is to predict if a protein is a beta barrel, and this one is for to predict if a protein is a membrane proteins. So since it in this Busca integrate a lot of tools, um, the prediction time is quite long. But the benefit is that the information it provides is rich. So you can see, besides the localization prediction, it also annotated following features. It tells you where is the signal peptide and where is the target peptides. So target peptides for mitochondria and chloroplast. And also it tells you if it is a GPI anchor. And for membrane proteins, it also tells you which region is alpha or beta region. So since this it took a long time to predict, I actually have a results I have already predicted before, so we can directly see what they provide in their results. Yeah, so this is five example proteins provided by their website. So you can load the example faster here. So this is a very good functionality. And this first you can roughly see what's the predicted geo term and what's the score. I think this can be used as a confidence, and some other alternative localizations and the predicted features. So we can look at this second one, since it predicted to possess have these signal peptides and the transmembrane alpha helix, and it will test you which region is the signal peptides. So it tells you from amino acid 1 to amino acid 24. That's the region for signal peptides. And this row is for transmembrane alpha helix. And it tells you that from amino acid 560 to 581, this region is for the alpha helix. So I think this is very useful. So besides the localization prediction, it actually tells you a lot of more. And similarly for the other things. For example, this one is predicted as a chloroplast protein, and it tells you the target peptides for the chloroplast protein. So it lasts this long, and it is also transmembrane proteins, and I think this one pass this membrane multiple times, so it will has multiple regions for the transmembrane alpha helix. So that's the main uh, the prediction results provided by Busca. Okay, we can move on to the next one. So the first ones are localization terms that you can just select it based on your interest. For example, you can select 10 categories or 5 or 3. But there's another way to define the localization targets. That is based on the GO hierarchical structure. And here is a tool to predict actually this tool is for protein functional prediction, but since CC, which is a cellular component, is also one of the GO category, so it also provides this CC prediction, which is we can use that as the protein localization. We can and this is actually a deep learning method. Here is a structure of this method. So the top ones are very straightforward. They just encode these protein sequences into an embedding layer. The way they encode this is based on the continuous 3 amino acids. And they will go through 1D convolutional layer to further reduce the embedding size. And I think one unique feature of these methods is that they retain this hierarchical geo architecture in their deep learning architecture. So you can see for each geo term, they developed its own fully connected layer. So since the geo terms have this children and father structure, so they keep that structure in this deep learning method structure. And they finally concatenate all the results from each GO term and output this final output vector. So you will know if it is predicted for each GO term. So that's the basic idea of this method. And we can go to their web server tool to have a look. So this method is called DeepGo. And they later they have another paper called DeepGo Plus. It's actually an upgrade of the DeepGo method, but the upgrade is minor, so they only like extend the acceptable protein sequence length and some minor changes. So the basic idea is the same. So the input is also very straightforward. They accept the protein sequences. You can paste them here. But there's one thing, one issue I noticed that is I will show you later. So we can just submit one example protein sequence they provide. So the issue I mentioned about is they actually ask you to determine the predicted threshold, and you can see if you select as 0.3, it will give you a lot of prediction results. So some of them are not corrected. Maybe they have a very low threshold. But I also checked this. Even the top ones are not very useful. So I think this thing is very not good for the users because for regular users they don't have idea how to choose this threshold. Maybe choose too high will result in none of the results is predicted. So anyway, the so you can see besides this cellular components, it also provides the prediction for molecular function and a biological process, and they will keep that structure. That means if you predict this protein belongs to one sub node in that structure tree, the structure, and it's not tree, structure graph, it will also automatically predict all the parents for that node. So that's the reason it will always have a very long list of prediction results. But the point is that I think it's also very unique is that we can think about this protein localization from the points from the perspective of GO. So basically, and one benefit is that it also reached a very detailed level. So we always talk about the subcellular, suborganelle. So in GEOs, it will have very detailed resolution for the localization. So I think this is the one good thing for these tools. And this is the recent work. It's actually a series of papers. The first one is SIGWAG. So in this work, they represent the protein sequences as a continuous vector or embedding by using existed a large language model called MMO. And after they do this embedding, they simply followed by a simple neural network, and they have two following tasks. One is protein structure prediction, and another is for 10 class protein localization prediction. And later, they didn't use the existed protein language model, but they train their own language models. They use several different deep learning architectures for the training, including two autoregressive models, that's Transformer XL and XLNet, and four autoencoder models, including BERT, Albert, Electro, and Transformer T5. And the data they use is from the UniRef and the BFD, containing up to 393 billion amino acids. So usually you want to train a language model, you will utilize a lot of data. So after they train their own language model specific for proteins, they again followed by a simple neural network to do the protein localization prediction, and the results is very good. So the collection of all these protein language models, they call it ProdTrans. And most recently, they developed this light attention model. You can consider it as a deep learning layer. So again, they use this large language model from the ProdTrans and replace this simple neural network with the light attention layer, and they found that the performance increased further. So in their paper, they also compared the different combinations using different language models, and they found that using ProdTrans T5 achieved the best performance. So that's the reason they in their web server tool, they only provided the model from ProdTrans T5. So this is this is by default, and you cannot choose others since this is the best performance. And you can just select one. You to accept a different format. usually you can see from the previous tools, they only accept the facet A, but in this one, you can also give a UniProt session and other IDs. So in this case, we just test a facet A format, and you can it directly, since I think this one is already in their database, so you can directly see the prediction results here. I think the information they provide is a lot. So first, you will know the subcellular localization is a mitochondrion. Also tells you it is soluble or not. Also, it will tell you some geo terms and with their confidence score. And you can also open the geo graph by clicking this button. And it also, besides this protein level prediction, it also provides some residual level features, because since their method utilize the transformer, so they retain the information or the attention from each residue. So you can see which region is this disorder region, and you can also see the transmembrane region. So this one is helix region, and this is a beta sheet regions, and some other information here. And also you can see the secondary structure prediction and the secondary structure in 8 states. So this is 3 states, and this is disorder prediction. So I think this is a very nice work. It actually opened the area of using the big large language model in protein localization prediction tools in deep learning methods. So later we will see some other methods utilize their large language model. That's another thing besides this prediction tool. One another contribution of this work is that they provide their source code and trained large language models. So you can just view by their GitHub, and you can get their code and download the well-trained large language models. So you can see their productivity file XL is here. So you can just download it and utilize it in your deep learning architecture instead of train a language model by yourself. So this is a very useful resources.